Hello, this is Professor John Glasgow, Dr. John, here to discuss the rubric for the journal article critique, which is your summation assignment for PSC 5031 research methods. The objective behind this assignment is to demonstrate what you've learned from the reading and from the course material. If you take a look at the rubric for the journal article critique, which is posted in the resource section, note also as well the instructions for the journal article critique, which are posted as well in the resource section. You'll see that I have some requirements there that are not APA. It's a slightly different opening. You do need a cover sheet, though. Please have a cover sheet with a proper running header. And do take a look at that. Looking at the rubric for the journal article critique, it might be a good idea for you to open it in split screen right now. I have it open on another monitor, so I'll be glancing off to my side to go over each one of these points with you. The very first point is that if it's on time, you get 8 points. Please submit it to Sakai. I have about 300 emails in my inbox every day. And if you submit assignments through email, I will certainly try to accommodate them. However, they can get lost. I tend to lose things on a regular basis, it seems. Okay, take a look at the rubric for the journal article critique that is found in the resource section. The first thing is 8 points for being on time. Second, APA format, professionalism of presentation, no proofread errors. It's amazing how many papers I do get with spelling errors that are highlighted by Word and that people haven't picked up. If you have proofread errors in there, that demonstrates a lack of professionalism. It isn't something that you would want to present to your clients or to people you were working with, your colleagues, in a report. So make sure you don't have any proofread errors in there. Identify the hypothesis or multiple hypotheses if there are those. State the null hypothesis. The null hypothesis basically says that whatever the researcher is trying to prove does not work. And you know that the null hypothesis is a construct that we work with in statistics because it is easier to disprove something than it is to prove it. To prove something, you'd have to show that 100% of the subjects in your population conformed to the hypothesis that you had written. To disprove something, all you have to do is find one that does not conform. So identify the null hypothesis. An example would be, I hypothesize that short-term memory will improve subsequent to 10 sessions of guided mindful meditation. And of course, I would have to concretize my variables for that. But the null hypothesis in that case would be that guided mindful meditation has no effect on short-term memory. So you need to identify the hypothesis or hypotheses and state the null hypothesis. You won't find the null hypothesis written in your research article. You have to construct it based on the hypothesis they have. Identify your independent and dependent variables. It is ironic that the independent variable, thing you would think you would be leaving alone, is actually the variable that you manipulate. It's simply identified as the IV. The IV is the manipulation in the experiment. In the example that I just gave, the manipulation would be guided mindful meditation. It is what you do to make a change in your experimental group. The dependent variable is wherein you measure the results of your intervention. And hopefully, if you are hypothesizing that guided mindful meditation improves short-term memory, your dependent variable would be a change in short-term memory. In your research critique, you're going to have to identify the independent and dependent variables. You're going to have to state whether they are sufficiently concretized or not. That simply means whether or not they have operational definitions. In the example I gave, if I simply said guided mindful meditation improves short-term memory, it's not sufficiently concretized because no one could duplicate that from simply the words guided mindful meditation. Now, how could you provide enough concrete information to make that duplicable? Well, you could provide a video of the person who was doing the guided mindful meditation so that someone else could reproduce it exactly, or perhaps someone else could use the actual video. That would be even better. That would be a concrete definition, concretely defined independent variable. The dependent variable there would have to be defined in some manner. Improvement in short-term memory, you might say, as demonstrated by a web-based letter recall test, and those are commonly available on the internet. You could give the URL for that test, and that would be sufficiently concretized. Then in that way, the testable hypotheses would read something like this. I hypothesize that 10 sessions of guided mindful meditation, as per the attached video by Dr. John Galaska, will change short-term memory as measured by results on a post-test compared to a pre-test of letter recall on a web-based short-term memory test, and you could give the URL for that. That would be a sufficiently concretized, testable hypothesis. Someone else could duplicate it if they wanted to. Going down further, you see describe the sample and how derived. The best way to get a representative sample is a truly random sample. If I were to just take, for instance, our class and use it as a sample in some sort of research, that would be a convenience sample, not randomized at all, might not be representative of the population to which I would want to generalize my results. Just a note on the side here, how many of our research articles are based upon Freshman 101 who are required to participate to gain credit for the course in a research project? I think you may find that we have a significant amount of our science based upon a population which may not be representative of the general population. The sample being Freshman 101 in a 101 psych class. Blinding. Oh, and of course, give the number of subjects in the sample. You know that in order to assume normality, the minimum number is going to be 16. A sample, an ideal sample size is often thought of as about 30. The ability to reach statistical significance increases greatly when a sample reaches about 16. And then it increases gradually up to about 100. And after that, not much more. So any number between 16 and 100 is probably good. 30 is often thought of as ideal for statistical analysis. Keep that in mind. You might mention it. Some pilot studies especially have very small samples, mention the difficulty of assuming normality with a small sample, and mention the problem with the sample perhaps not being random. And of course, if a sample is not representative, you have difficulties with external validity, generalizing to a greater population. Blinding. Talk about blinding even if the study does not mention it. If they do not mention blinding, report the lack of blinding. Blinding of the research subjects, not knowing whether they are in the experimental condition or the control group, is one level of blinding. If the researcher himself or herself does not know which subjects are going to be placed into which group, then you have what's called a double blind. If the person who is measuring the results, scoring the tests or whatever they are to determine the effect on the dependent variable, if that person is blinded as to the assignment of whether the subjects were in the experimental group or the control group, then you have a triple blind. Randomized, double blind at least, triple-blind studies are the gold standard for research. This may change in the future. You may get to see that change. Stay tuned. Okay, control group, contrast group, or groups identified and how are they formed. If you have a group of 30 and you put 15 of the subjects into a wait list, that's a really neat way to do a study, as long as there isn't any pathology that might deteriorate or worsen the condition of the subjects over the wait list period of time. It's a very convenient way to take a number of subjects and split them. If you have 32 in your group and you split them 16 and 16, then you will have a minimum in your control group and experimental group, that n having been split in half to 16 to assume normality. So how was the control group or the contrast group formed? Mention that. Suggest possible confounds, uncontrolled variables, and bias. In bias, it's always good to mention who's paying for the study. If you're looking at a study about baby powder and Johnson & Johnson is paying for it, you might suspect that the results might be slightly tainted by the researchers that they have selected to perform that study, since there's literally millions of dollars involved, and that tends to color the perceptions of the people doing the research. You know that back when tobacco was being called into question, there were lots of researchers who came out and said, hey, my gosh, tobacco is not really bad for you. And guess who? Probably Philip Morris or one of the large tobacco giants funded those studies. So look to see who's paying for it. That is always a possibility of a conflict of interest. Notice if the researcher is the one who actually assigns the subjects to the control group or the experimental group, you have the potential for researcher bias. If the subjects in the research, the people who are participating, are aware of which group they're in, and they know the intent of the research, and some deception is not out of the question in research. If the researcher is the one who places them, the researcher could have bias in placing them. If the subjects themselves know which group they've been placed into, they may, especially if they are volunteers, want the researcher to be able to prove this hypothesis. Then you have what's called participant bias. So mention the possibilities of researcher and participant bias. In some cases, you may see that it's pretty blatant. So also suggest possible confounds. Things, for instance, like the conditions under which the original pretest was done varied significantly from the conditions under which the post-test was performed. If you tested subjects in the morning in the pretest, tested subjects in the evening on the post-test, that's a confound. Time of day should be the same. All controllable variables should be the same. The idea is to make everything as close to the same in pre and post, with the only difference being your manipulation. Okay, that's the ideal. Now, it's impossible to control all the variables. But sometimes there are blatant things that are left uncontrolled. Morning, evening, temperature of the room, stuff like that. Okay, mention it. Those are possible confounds. They would affect internal reliability, which of course is necessary if you want to have generalization or external validity. Compare the article to another source. Somewhere in your conclusion or your discussion, you should find another article that talks about exactly not exactly the same thing, but something very similar to what this article addressed. And you should mention their criteria. And you might say something like, Jones and Jones came up with a similar conclusion with a sample of 42 college students in 1999. Brief mention, you can often find articles cited in your research article that are of previous research. You could use one of those, but do bring one in at least. And that added source has to be cited. Describe your research design. Is it within subjects, between subjects? And of course, within and between subjects have synonyms and statistics just to make things a little more complicated for you. Within subjects is often called dependent measures, and between subjects are sometimes called independent measures. Those are synonyms. The kind of statistical analysis that you use will depend upon whether your design is within or between subjects. In some cases, you may have a factorial design or other research design. Next item, describe the utility or lack of it for personal use in the mental health field. This means for you. And I had hoped that most of you would be able to find an article that falls into your area of interest so that you could find something that would be of secondary value to you going into the field. So whatever the article discussed, mention whether you think it will be utilitarian for you or not. Okay, don't be afraid to take a negative position in your eventual practice. So you're also going to be graded on the proper use of research and statistical terms. In some very rare cases, I have found, I have read papers wherein students used terms with absolutely no idea what they meant, in sort of a blind hope that the actual use of the term in a sentence, even if it was used incorrectly, would somehow suffice to demonstrate understanding. That doesn't cut it. So don't be guilty of that. Know what you're talking about, know the terms. Use as many of the terms as have been presented in the reading, in the course material as you can, and use them properly in your critique. You must mention reliability and validity of the instruments used. If you're doing a short-term memory test, you need to find out whether that test has psychometric data associated with it. If it does not, mention that. Short-term memory test that we had has not been researched. It was made up by the researchers themselves, and therefore there's no reliability or validity information available on it. That could be the case. However, in most cases, you're going to find that your researchers are going to mention that they've used a test that is a standard in the field. For instance, they might say, we use the Beck's depression inventory pre and post to measure depth of depression. Beck's depression inventory has been shown to be reliable and valid, and you might give a reference for that. In the resource section of Sakai, you'll find a short topic. It says reliability and validity of research instruments or tests. I'm not sure exactly how it's worded, but it starts off with reliability and validity. Look at that 3-minute video. It will give you clues on where to find psychometric data for the tests that are utilized in whatever research article you've got. Okay, discuss generalizability. That's internal and external validity. If you have internal validity, if your procedures were valid and reliable, and they were properly done, you have internal validity. That's the first step in being able to generalize. Second, you have to have a representative sample that is large enough to assume normality. If your sample is representative and you have internal validity, then you have generalizability. That is the same as external validity. Okay, another case where in statistics we use several terms to demonstrate or to signify exactly the same concept. So talk about generalizability. You might state something like, This sample that was drawn from a freshman class of students in Duquesne University is not representative of the population that they wish to generalize to. Or you may say, yes, it is, and state your reasons. But discuss internal validity, external validity, and generalizability. Discuss both positive and negative points in this study. There will be some things that will look really good. There will be some things that you may look at and feel that they are actually, they detract from the quality of the study. Mention both, okay, please. Duplicability. Was enough information presented in this study so that someone else could look at it and duplicate the study? That would entail having operational definitions for the independent and dependent variables, having very clear explanations of the processes that they used internal to the study. Duplicability is a big deal. If your study demonstrates something of importance to the field, you can bet there are people out there who are going to try to duplicate those results. So they're going to want to look at your study and try to do it exactly the same way you did with another sample and see if the results actually confirm yours or deny it. Science is a publicly debated, publicly entertained, and open topic. And so research that is presented it's an invite for duplicability. Can your research topic be duplicated? Suggested further research with alternative designs, methods, samples, and/or hypothesis. You might say that, oh my goodness, we looked at this and we found out that there are other factors that may be more important, and that is often the way research goes. Things don't often go exactly as you planned. So based on that, even if your research is negative or the article was unable to prove the hypothesis, prove causality to the level of significance that they had hoped, you still have learned something. And further research could be suggested with perhaps an alternative design, a different sample, different methods. Do that. That's a required part here. Now, a general category down at the bottom: clarity of ideas, organization of thoughts, and use of graduate level English. Has to be grammatically correct. It should be well expressed. Avoid very long, complicated sentences unless you can make it very, very clear how all of the ideas in your long, complex sentences are related to each other. I have found that in reading research papers that sometimes students get lost with long sentences. Often it is better to split those up into two separate ideas. It's okay to have long sentences that are complex as long as the ideas are very clearly related to each other, and that relationship is very clear to the reader from what you've written. Generally speaking, myself, for my own style of writing, I like to use some long sentences, and then I like to put in a little staccato, 3-4 word sentence, ta 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 ta. It gives, I think, some relief to the reader in terms of trying to follow what's going on, and it's my own personal style of doing things. That's the way I like to do it. You may find it of value as well. I hope you find this video of interest and take a look at the instructions for the journal article critique that are in the resource section. Do follow the suggested format that I have. Submit your paper on time to Sakai. If the window closes and you submit it for email, I will accept it. However, I will ding your grade somewhat for being late. And sometimes I lose emails. I get so many of them, especially if they are attached to other emails. That's one sure way to make your email get lost. Instead of attaching something to a previous email, it's best to start a new topic, stating very clearly in the subject what it is you're doing, like assignment number 6, ta 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 ta, and then state your reasons why it's submitted late. Um, I really don't want a detailed explanation of what's going on in your life. I trust you. If you have mitigating circumstances, just let me know. At any rate, good luck with this. This is the summations project for this course. It's the opportunity for you to demonstrate what you have learned. And you can use this format for future journal article critiques. Of course, do whatever your professors want, but journal article critiques will be a part of your ongoing academic experience. This is Dr. John, and thanks for listening.